Hello and welcome to the screencast video for the urinary system. Without further ado, let's just get down to it. So first thing, just basic overview of what the kidney does. And most of you are probably familiar with the fact that yes, it filters the blood, but because it is a site where blood is processed, a lot goes on there, and it does a lot more than just excrete stuff. So there is, as this says here, removal of exogenous chemicals. That just means stuff that doesn't necessarily belong in the body. It might be drugs. It it might be an additive in your food that's non-nutritive, whatever it is. Generally, stuff that you end up eating that ends up in your bloodstream, your kidney helps get rid of it. Metabolic byproducts. This is mostly nitrogen from amino acids that come from proteins that you burned for energy as opposed to using to make your own proteins, and then excess ions. Because your kidneys are able to process the blood and affect how much sodium, here we go, sodium and water is in the blood, they help regulate blood volume, which then affects blood pressure. I think we've hit blood volume, blood pressure a couple of times. It's one of the central themes to anatomy and physiology too, because blood pressure is one of your most important bodily variables. Then also, because your kidney processes the blood and has the ability to control or pump, I should say, things like hydrogen ions and bicarbonate ions, your kidney plays an important role in maintaining proper blood pH. So just to give you a heads up of how the chapter is going to work, we're going to start with basic anatomy, just run over the parts of the kidney. That'll be really simple. Then we're going to go over, it's a gross oversimplification of how urine is made, the basics of the physiology. Then we'll go into more detail about what happens every step along the way, going from blood to urine. So we're going to sort of keep drilling down. Just to start with the anatomy, you're not going to have to know this for lecture exams, but we need to know all of this terminology before we can talk about how the kidney works. So I'm not throwing out terms you haven't heard before. Where am I looking for? Oh, this says renal artery here. It should be pointing like right here, behind this renal nerve, which has no business being in that picture because we don't care about the nerve. But whatever, blood's going to come in the renal artery, exit the renal vein. We'll talk about the filtration process later. We'll start on the outside. So just like the outside of your brain is your cerebral cortex, the outside of your kidney is your renal cortex. So this whole outer region is cortex. This sort of middle region here is called the medulla, as it's said over there. It is divided into individual structures called pyramids, but we don't really care about that right now. Then in the middle is the renal pelvis, and this is where all of the processed urine drains. So for us, we want to know cortex versus medulla is going to be important, and then know that the urine ends up in the pelvis. If we look at blood flow, again, this is just overview. Oh, this picture is not as big as I thought it would be, but who cares? Blood is going to flow in through your renal artery, and then it goes through smaller arteries, which you will only need to know the name of in lab, which then run out into the cortex. In the cortex is where all of the nephrons are located. So that messy little structure that looks like spaghetti with blood vessels on it is a nephron. Each of your kidneys contains a few million of them. So you can think of your kidney as containing a million different filters that all run in parallel. Right, so blood is going to be sent to a nephron. stuff is going to happen. Blood flows through all of these capillaries, comes out the other side, and then is drained. So this filtered blood will then drain into venules, bigger veins, and out the renal vein the same way the renal artery came in. So blood goes in this side of the nephron, urine comes out that side, and then the urine hopefully ends up in a proper receptacle. Um, Looking closer now at the nephrons, this is the structural unit of the kidney. Um, as it says, about 1 million per kidney here. There are two sets of fluid handling tubes. You have tubules, which are going to process the filtrate, which will eventually become urine, and then the capillary beds. You want to think of them as parallel fluid processing lines, and in a couple of slides, we'll make a little bit more sense of that. Um, so first we're going to talk about the tubules, then we're going to talk about the capillary beds. There are three different parts to the nephron, or three different kinds of tubules. You start here with the glomerular capsule. This is going to collect fluid that leaks out of these blood vessels here. That fluid will then flow through first a proximal convoluted tubule, then down the descending limb of the loop of the nephron. My slides still call it the loop of Henle because I'm stupid and I forgot to change it. So down the descending limb, up the ascending limb of the loop, and then into what is called the distal convoluted tubule, which then drains into a collecting duct. And you can see here, 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 and there, those are the distal convoluted tubules of other nephrons that are all feeding into that collecting duct. And then that collecting duct runs through the medulla and drains into the renal pelvis. And which slide were we on? So then the capillary beds, there are going to be three different vascular structures or capillary beds that we care about. The first is the glomerulus. It is this tangle of blood vessels here, which is surrounded by the renal capsule, or what used to be called Bowman's capsule. Blood flows into it through an afferent arteriole. Fluid leaks out of it. That fluid we are going to call filtrate. And then blood leaves the glomerulus via the efferent or efferent arteriole. Again, this is where filtration takes place, and the blood pressure in this capillary is relatively high. That encourages fluid to leak out. And we'll talk more about filtration and all of the steps as we go. Right now, we're just reviewing anatomy, or introducing anatomy, I should say. So then you have a group of capillaries that are close to your proximal and distal convoluted tubule, so we call them the peritubular capillaries. These have low pressure, and they're porous, which means it's easy. They are, as it says here, they are adapted for absorption. You are going to see that stuff in the tubules ends up being transported back into the bloodstream. So you want plenty of room in your tubules, low pressure, so it's easy to get salt and water back into your bloodstream. It's also important to point out that these peritubular capillaries arise from your efferent arteriole. So you want to think of the blood losing fluid in the glomerulus, or in the capsule, and then the bloodstream flows through the peritubular capillaries parallel to the fluid that it just lost. Then you have just another group of blood vessels also connected to the peritubular capillaries called the vasa recta. And all you want to know right now is that the vasa recta is closely associated with the loop of Henle, or the loop of the nephron. So now we are into sort of going step by step what happens in each different part of the nephron. This is going to go a little bit slower and will be more detailed, and this is where we're going to be for quite some time. So we start with step one, which is the formation of filtrate. All right, so here's how filtration works. As I mentioned earlier, blood is going to flow in through what is called the afferent arteriole. Then it enters this tangle of blood vessels here called the glomerulus. Um, Fluid leaks out because the blood in these blood vessels is under pressure, and those blood vessels are porous, so fluid leaks out. This filtrate is going to flow through the rest of the nephron. That is going to become your urine. So the next thing you want to understand about filtration is the structure of the filtration membrane. The most important thing to remember for the exam is simply just the parts of the filtration membrane. So you start, if you look at just the glomerulus itself, you have blood vessels that are covered by cells called podocytes. And the way the structure works is if we start from the inside, the cells lining that capillary, that endothelium, are highly porous cells that have little holes in them, actual holes in the membrane, called fenestra. So it is called a fenestrated endothelium. That makes this cell or this blood vessel here extremely leaky. But then surrounding it is a basement membrane, um, which we'll go over in the next slide a little bit. But this you are going to see is actually more restrictive than the fenestra. So it has really, really, really tiny pores in it that are smaller than these little pinprick holes you see in the endothelium. Surrounding the blood vessel, again, are these cells called podocytes. I don't ask questions about them. Well, you do want to know that they're there. I take that back. You want to know that the podocytes are there. You don't need to be able to describe foot processes and filtration slits. But to understand how it works a little bit better, you want to know that they're there. Basically, fluid can't flow directly through the podocyte. It does resist fluid flowing through it. So most of the filtration happens where the podocyte foot processes interdigitate with each other, and those are called the filtration slits. So the most important thing that you want to know is that it is the basement membrane that ultimately decides what ends up in the filtrate and what stays in the bloodstream. And it is a simple size restriction process. So anything smaller than a protein will leak out of the glomerulus or out of the capillaries and into the nephron. That is what is going to form the filtrate. So this means that the filtrate, what is ultimately going to become urine, contains basically anything that you might find in the plasma minus the proteins. That means there's lots of water in the filtrate and lots of stuff that you want: glucose, vitamins, ions, hormones, you name it. If it's in the plasma and it's smaller than a protein, it is in the filtrate. There's also stuff in the filtrate that you don't want, like urea, excess solutes, like more sodium, or excess potassium. So what your kidneys have to do is then sort through everything that's in the filtrate, take all the stuff that you do want and pump it back into the bloodstream, and then leave all the stuff that you don't want in the filtrate so that that ends up being excreted. So what actually comes out of your kidneys represents about 1% of what leaks out of your glomerulus. All right, so now that we have a sense of what's in the filtrate, I just want to go over quickly how your kidneys go from unprocessed filtrate to processed urine. So as we said, if you're going to make urine, step one is glomerular filtration. So anything smaller than a protein leaks out of the glomerulus into the capsule and then is going to flow through your tubule system. So this filtrate is going to contain some stuff that you want to keep, like sodium and water, and then all of the waste products that your body wants to excrete. So as the filtrate is flowing through the glomerulus, one of the things that happens is called tubular reabsorption, where stuff like the sodium that you want to keep is pumped back into your bloodstream. If the sodium is pumped back into your bloodstream, this creates an osmotic gradient, and then water is going to follow. So reabsorption includes both solutes and water. And then tubular secretion means that what your body is going to do is take waste products or anything that it doesn't want to keep and actively pump that into the filtrate, so that you can concentrate waste products in the filtrate with a minimum of water and sodium loss. So most of the water reabsorption, the bulk of this, ends up being pumped back into your bloodstream early on in the filtrate processing portion. But if you are dehydrated, at the end in your collecting duct, your kidneys can pull last couple percentages of water back out of the filtrate. All right, so we're still actually talking about filtration. We're going to spend quite a few minutes just talking about this very first step, because it's the most important step. You're going to see your kidney puts a lot of effort into controlling this. And we'll talk about why in a minute. Let me just move on. So the first thing we want to understand or know is just this term up here, your glomerular filtration rate, or GFR. That just means how much fluid is leaking out of all of your glomeruli and all of your nephrons. For us, what we care about the most is the net filtration pressure, or the blood pressure in the capillaries of your glomerulus. There are other factors that affect the glomerular filtration rate. They are shown here, but we don't really care about them. We've got enough to learn without going into every nitty-gritty little detail. So just looking at this right here, I want to give you a little bit of perspective, right? This is all of the stuff that needs to be either pumped out of or pumped into the tubules as filtrate is being formed. So there are lots of different pumps and channels and processes, and it's really complicated. So what your body does is try and control step one very closely and keep your filtration rate relatively constant so that all of these other processes can run on autopilot. So if the amount of filtrate that's being formed is always the same, then all of these processes of pumping this and that and the other thing can all run at a constant speed, and you don't have to constantly adjust all of these different pumps and mechanisms here. So we are going to be right here, anatomically speaking, for a little while, talking about the different mechanisms that govern the filtration rate, because that's the process that your body puts the most effort into controlling. So before we go into the processes, we have to understand the anatomy. So we're going to introduce more terms and names of cells and stuff. The first thing you want to be familiar with is just this term here, the juxtaglomerular apparatus, which just refers to all of this crap here, which is close to the glomerulus. The parts of the juxtaglomerular apparatus that we care the most about are the macula densa, highlighted in green right here. This is a group of cells that are in part of your distal convoluted tubule, or the very top of the ascending limb of your loop of the nephron. Then there are these cells right here called juxtaglomerular cells. They're also sometimes called granular cells. They have a couple of different names. And then the afferent arteriole. What we're going to be focusing on primarily is the role of the afferent arteriole and the macula densa in regulating the glomerular filtration rate. So we are going to have two different kinds of mechanisms that affect glomerular filtration rate. Intrinsic controls. So these are controls, as this says right here, act within the kidney. So they don't respond to variables outside of the kidney. And there are going to be two extrinsic controls, your myogenic response and what is called tubuloglomerular feedback. Then extrinsic controls are when your kidney is being affected by stuff outside of your kidney, like overall blood pressure, your sympathetic system, or different hormones. And we'll go into all of that. So first up are the intrinsic controls. The way the intrinsic controls work is that they are designed to maintain a constant glomerular filtration rate. So you're sort of assuming that your body's overall physiology, blood volume, blood pressure, pH, and all of that is relatively normal. So nothing drastic has to happen in the kidney to maintain homeostasis. So this is where your kidney is trying to keep your filtration rate constant, so all of the tubular reabsorption and tubular secretion can run at a constant rate. Both of our intrinsic control mechanisms are going to work by causing either vasoconstriction or vasodilation of the afferent arteriole. So you're controlling how much blood goes into the glomerulus and therefore the blood pressure in the glomerulus. And that's what determines how much fluid leaks out in the glomerulus versus stays in the glomerulus. So first up then is the myogenic response. And remember, the name of the game here is to keep your filtration rate the same. So the myogenic response is governed by the afferent arteriole. It has the ability to sense and respond to blood pressure. So the smooth muscle in your blood vessels, or smooth muscle everywhere, is really special and has lots of abilities that skeletal muscle and cardiac muscle don't have. So if your blood pressure goes up, what would normally happen then is that more blood would flow into the glomerulus, and that would cause an increase in your filtration rate, which your kidney would then have to adjust to. What your afferent arteriole does is when blood pressure goes up, it constricts so that blood pressure in the glomerulus stays the same and your filtration rate stays the same. Conversely, if your blood pressure dropped, less blood would flow into the glomerulus, the pressure in your glomerulus would drop, and your filtration rate would drop. So when your blood pressure does drop, your afferent arteriole is going to vasodilate and allow more blood into the glomerulus to try and maintain the blood pressure in the glomerulus, which would then again keep your filtration rate the same. So that's the myogenic response. Then you have what is called tubuloglomerular feedback. This is a little bit more complicated to understand. I shall do my best to explain it to you. The first thing you want to know is that this is controlled by the macula densa cells, which, as we said, are these little cells right here in green, located in the top of your ascending limb or the beginning of your distal convoluted tubule. What you want to know is that by the time filtrate gets to your distal convoluted tubule, it is largely done being processed. It is close to a finished product. And so what your macula densa really is kind of a quality control check towards the end of your urine formation process. What the macula densa senses is sodium levels. So it uses sodium as a measure of how completely processed the filtrate is. So under normal circumstances, your body holds on to as much sodium as it can, or most of the sodium, which means sodium levels in the distal convoluted tubule should be relatively low. And I forgot the way they have this drawn here. It looks like the distal convoluted tubule is far away from the glomerulus, but in actuality, most of your nephrons, the distal convoluted tubule is right next to the glomerulus. So you have a quality control checkpoint towards the end of your processing line, which is actually geographically located very close to the very beginning, right at the very first step. So your macula densa can track what's coming out the end of the nephron and is located right next to the beginning of the nephron, so it can affect that first filtration step. So then, how does it work? Bearing in mind that it is also trying to match your filtration rate to your tubular reabsorption and tubular secretion so that your pumps and channels in the tubules can run on autopilot, and what is adjusted is your glomerular filtration rate or the pressure in your glomerulus. So let's say for whatever reason your GFR has gotten too high, or your kidneys need more time processing the filtrate. So if your filtrate is flowing faster than it's supposed to, or faster than your nephron can handle, this means that there is less time for sodium to be pumped out. If there's less time for sodium to be pumped out of the filtrate, that means that sodium levels in the filtrate are going to be high when the filtrate reaches the macula densa. If the macula densa senses increases in sodium levels, it is going to release adenosine triphosphate. So here's your macula densa. If there's a lot of sodium in your distal convoluted tubule, it releases ATP, and that is going to cause your afferent arteriole to constrict, and that will reduce the blood flow into your afferent arteriole. So if too much is coming out here, that's what my big goofy arrow is supposed to mean. You get vasoconstriction, so less goes in here. And then if you have less going in here, you have less coming out there, so that you can properly match your filtration rate to your filtrate processing speed. Conversely, if your glomerular filtration rate is too low, this means filtrate is going to be flowing very slowly through your tubules. They will have more time than they need to process the filtrate, and they'll end up pumping out more sodium than they normally would. That would cause your sodium levels to become abnormally low. So here we have the small arrow, which means slow flow. That means that by the time the filtrate gets to the macula densa in the top of your ascending limb, the sodium levels are now very small. This causes the macula densa to stop releasing ATP, and in the absence of ATP, your afferent arteriole will vasodilate. That lets more blood into the glomerulus, which is going to bump your glomerular filtration rate up. Both of those processes are about either keeping your glomerular filtration rate constant or matching it to the speed at which your kidney is processing things. Then we have your extrinsic controls. So this is where conditions outside the kidney are going to affect how the kidney works. And there's nothing important on this slide here. It just sort of sets the stage. Let's go to the next one. So the first of the extrinsic controls is a little complicated, which means I think it's fun. You may or may not think so. This is called the renin-angiotensin mechanism. So it is generally triggered when blood pressure is very low. That drop in blood pressure is going to cause your granular cells, or what I just called your juxtaglomerular cells, to release renin. Renin, or your granular cells, can also be stimulated directly by the macula densa or your sympathetic nervous system, but we're primarily going to think of renin release as being governed by blood pressure. So a drop in blood pressure means your granular cells, or these cells right here called juxtaglomerular cells, are going to release a hormone that's called renin. So the way it works is that your liver produces a prohormone, or an inactive hormone, not hormone, enzyme, sorry, and called angiotensinogen. So it is always floating around in the bloodstream, ready to jump into action when your blood pressure gets low. The signal to angiotensinogen to jump into action is renin. So renin is going to convert angiotensinogen into angiotensin 1, which will then circulate everywhere throughout your body. Everywhere in your body where there are blood vessels, there is another enzyme called angiotensin converting enzyme, or ACE. It converts angiotensin 1 to angiotensin 2. This just ensures that because angiotensin 1 is going to go everywhere, it's in the bloodstream, having angiotensin converting enzyme embedded in the blood vessel walls makes sure that a little bit of angiotensin 2 gets produced everywhere, instead of having one source of angiotensin 2 where there's going to be a lot of it and then less of it as you get further away from that source. So this mechanism ensures that angiotensin 2 is created everywhere throughout the body in a relatively short period of time. So what's more important for us is knowing what angiotensin 2 does. So all of the effects of angiotensin 2 are intended, or what they all do is cause your blood pressure to go back up. So it's all about maintaining high enough blood pressure. So angiotensin 2 gets generated everywhere, and it causes your arterial smooth muscle to constrict. That vasoconstriction decreases your blood volume, not your blood volume, but your vasculature volume everywhere, which brings your pressure up. It's also going to cause vasoconstriction in your afferent arteriole, which is going to slow down your glomerular filtration rate, so you're going to produce less urine when your blood pressure is low. It also stimulates the reabsorption of sodium. It acts both directly on the kidney and on the adrenal cortex, causing the release of more aldosterone. And as we talked about when we covered aldosterone, if you're pulling more sodium out of your urine, you're also going to pull more water out of your urine. So that's concentrating your urine, reclaiming more of your water, maintaining your blood volume, and therefore maintaining your blood pressure. I don't know if you can hear my dog barking in the background, but I can. She's cute but annoying. At any rate, the third thing here that I have angiotensin 2 doing is stimulating your hypothalamus to release antidiuretic hormone, which is going to, via another mechanism, cause your kidneys to concentrate your urine more and reclaim more water from the filtrate. And it also makes you thirsty so that you drink and bring your blood volume and therefore blood pressure back up. Your sympathetic nervous system can also get in the game. So under conditions of physical stress or low blood pressure, or even psychological stress, I should say, your sympathetic nervous system can release epinephrine and norepinephrine, which is going to cause systemic constriction of your blood vessels, bringing your blood pressure back up. It can also cause the release of renin and the constriction of your afferent arteriole. So your sympathetic nervous system can constrict your afferent arterioles, reduce your glomerular filtration rate, and trigger the release of renin, which is going to cause all that other stuff to happen that we talked about that keeps your blood pressure high enough. I am going to stop here because I'm tired of talking and you're probably tired of listening. I will make a second screencast that finishes up the rest of this, but I feel like 35 minutes is long enough and it'll take forever for it to load onto YouTube. So that's it. I'll see you in part two.